And welcome to the first of the hopefully many videos on GCSE content for AQ8 Science. Starting off looking at biology, we're going to start off looking at cell structure. The specification reference is 4.1.1. We're going to be looking at eukaryotic and prokaryotic cells, animal and plant cells, cell specialization and differentiation, and finally microscopy, so using those calculations and also remembering that required practical and how to write the method. Okay. By looking at eukaryotic and prokaryotic cells. The eukaryotic cells are our animal and plant cells. Our prokaryotic cells are our bacterial cells. Okay, so looking first at the eukaryotic cells, we've got an animal cell at the top and a plant cell at the bottom. The key part to recognize here is that the DNA is enclosed within a nucleus. Okay, we've got that large black dot, that is our nucleus. Inside there are all of the chromosomes, all paired up, all nicely put together, all in one place. The other thing we've got is the mitochondria. So the mitochondria are the site of respiration where we make energy, or ATP, adenosine triphosphate, to provide the cell with all the energy that it needs so it can carry out its functions. Size-wise, we're looking at about 10 to 100 micrometers. So quite a big size difference. Animal cells typically smaller than the plant cells. Animals at the smaller end, plants at the larger end. Now our prokaryotic cells, I know size-wise drawing them, the prokaryotic cells looks a lot larger. However, in reality, it's a lot smaller. It's got a length of about 1 to 5 micrometers, so a lot smaller. You'll also notice that there is no nucleus within the prokaryotic cell. The DNA is either just loose as chromosomes within the cytoplasm, or is there as plasmid DNA, which are rings of DNA. They come in very handy when we look at genetic engineering and how we can produce insulin. Now, one thing I haven't drawn on the prokaryotic cell is a slime capsule. Some prokaryotes or bacterium will have a slime capsule around the outside of the cell wall, which gives them effectively 3 layers of protection against antibiotics or predators. Other thing they have finally is that flagellum at the end, a tail-like structure just to help them with movement. They are all single-celled organisms, so they will need to move by themselves, whereas obviously our eukaryotic cells are typically multicellular organisms. Okay, things that you might need to do for your exams is you might need to identify whether a cell is a eukaryote or a prokaryote. To do this, we're going to be looking at the cell. You're going to see whether the DNA is enclosed within the nucleus. If it is enclosed within the nucleus, we can assume that it is a eukaryotic cell. Okay, that is our main way of doing it. Labeling parts of the cell, we need to be able to draw and label the parts of the cell. You'll notice when I've drawn mine, I've drawn them in pencil, and when I've done my labeling, I have draw my lines. They're not arrows, they are lines. In biology, we only use lines. Arrows represent the movement of something or the transfer of something. We're also going to do a comparison of the sizes. So our prokaryote is 1 to 5 micrometers. Our eukaryote are between 10 to 100 micrometers, so much bigger in size. Quick note for the sizes, okay, and converting our units. We have 1 meter. Is our SI unit of length. Okay, 1 meter is made up of 100 centimeters. Each centimeter is 10 millimeters. Okay, so we've got 1,000 millimeters are in a meter. Within a millimeter, which I'm sure you've all seen on rulers, we've got micrometers. Now we can fit 1,000 micrometers into a millimeter. Hopefully giving you an idea how small a micrometer is. They are tiny. We really struggle to see them with the naked eye. You can see some cells that are particularly big, especially the very large ones you might be able to see without a microscope, but looking at any detail you do need a microscope. And we use that funny U, which is the Greek letter micron, for microscopes. You can also see it written MCM or written micrometers. Okay, and then going even smaller, we've got 1,000 nanometers are within a micrometer. We get down to nanometer size. We're getting down to atomic size, very small. And plant cells. So I'm going to keep the picture up of the eukaryotic cells because they are my animal and plant cells. The animal cell again at the top, plant cell still at the bottom. You will need to know the difference between them, identify whether something is an animal or a plant cell potentially, and also label and describe what each organelle is. The organelles are the little parts within the cell itself. So we've got the nucleus. All cells contain a nucleus, obviously except for the red blood cell, which doesn't contain one. The nucleus contains the genetic materials. This is all the information that is in the cell. We've also got the cytoplasm. This is where chemical reactions occur. It's that jelly-like substance. You struggle to see it generally within the cell, but it is there. It's where everything occurs and it holds everything in place. We've then got the cell membrane. This is what surrounds the cell and it controls what enters and leaves the cell. And then mitochondria, again, all eukaryotic cells, so animal and plant cells, contain the mitochondria. This is where respiration occurs. So whether it's going to be aerobic or anaerobic respiration, it happens within the mitochondria. Certain cells will have more mitochondria if they need to provide more energy, for example, muscle cells. We've then got ribosomes, which are the tiny little dots. Now they are the site of protein synthesis. So protein synthesis is making their own proteins. This is a lot of what a cell does. It will make its own proteins. Now these 5 are all present in animal and plant cells. We're now going to have a look at the organelles that are just present inside a plant cell. So we've got the chloroplast. Now the chloroplast contains chlorophyll. Got lots of claws in there. I know it's frustrating to try and remember them, but they are there. Okay, so chloroplast contain chlorophyll. This is where photosynthesis occurs. Now, chlorophyll give that plant the lovely green colour. That's why we see nice green leaves. You need lots of magnesium to make the chlorophyll so that the plants can be green. Photosynthesis obviously is plants making glucose and oxygen from carbon dioxide and water. It's the reverse of respiration. We get the vacuole, which is in the middle. Okay, so the vacuole is a large bit, contains cell sap, okay, which the plant can use to store food or store anything it needs to store in there. Also helps provide a bit of structure and strength to the cell as well. Finally, we've got the cell wall. So the cell wall is around the outside of the cell membrane. It doesn't stop things entering or leaving, but it does provide structure for the cell. Also helps provide a bit of protection and stops the cell collapsing if it gets too dehydrated. Now in plants, they are made of cellulose. Look at cell specialisation and differentiation. So specialisation is where they have that special job to perform, so they will all do their own particular function. Okay, so we'll look first at sperm cells. Okay, so sperm cells are the male gametes, those sex cells. They contain half the number of chromosomes. Now they've got a variety of adaptations to help them with this. So depending on the species, they have different distances to move. They've either got to swim through water to reach the eggs that have been laid by the female, or they have to swim through the female reproductive system. Regardless, they have to swim, so they have a long tail and lots of mitochondria to provide them with lots of energy for this. They also have something called an acrosome, okay, which is that cap at the end, contains lots of enzymes to help them break through the egg. It's also got a very large nucleus which contains all the genetic information to be passed on. Obviously noting that it's half the number of normal chromosomes of a cell. It is a haploid cell, it contains half the number of normal chromosomes. Next up, we've got the nerve cell. So nerve cells carry those electrical impulses around the body of an animal. So they provide a really, really quick communication. So they've got several adaptations to do this. They've got lots of dendrites, and there's those bits at the end to make connections to other nerve cells. They've got an axon, that's what carries the nerve impulse from one place to another. It's that very, very long part of it. Can be up to 25 meters long, which is in a blue whale. With us, our longest one runs from the base of our spine to your big toe, so we're looking about a meter in length. Okay, we've also got a myelin sheath around the axon for insulation. This helps speed up the nerve impulse. And we've got synapses at the end that will send lots of chemical messages across the synapses. Okay, we've got lots and lots of mitochondria in there as well to provide that energy to do it quickly and to make those transmitter chemicals. Next up, we've got the muscle cells. So muscles are special cells that can contract and relax. Okay, muscles work in pairs. One will contract, the other one, if we think about our bicep, it will contract. Our tricep will then relax when we're changing our arm back the other way. Our bicep will relax and our tricep will contract. I've just realized that I'm doing this in a demonstration, but none of you will be able to see that. Very sorry for that. Okay, mitochondria or muscle cells are very, very special. So they've got a few adaptations. They've got special proteins. That means they can slide over each other. It means they can contract very, very easily. They can contain lots and lots of mitochondria. Again, this can provide them with lots of energy. They can also store glycogen. So this is what glucose can be stored as when the levels are too high in the body. Stored as glycogen, it's kept in the muscles, meaning when it's broken down, it is right by those muscle cells ready to use straight away. Finally, we've got those red blood cells. So the red blood cells are the cells that carry oxygen around our body. They contain lots of hemoglobin. There's no nucleus within them, so there's more space for hemoglobin, and they are a biconcave disc. Okay, they're a bit like a flattened donut. Okay, they've got that flat bit in the middle. This increases their surface area to volume ratio, meaning they've got more space to carry oxygen. Next up, we're going to look at plant cells. So within plant cells, again, we've got some adaptations. We've got that root hair cell. Now the root hair cells they're in the root, as we can tell from the name there. They've got this hair, they've got a bit sticking out of them. Okay, that is so that they can increase their surface area. This means that they can take in more water through osmosis and mineral ions through active transport. They've got that very, very large surface area. They've also got a large permanent vacuole, and this helps speed up osmosis through the cell. And they've also got lots and lots of mitochondria that can provide the energy for active transport. Now, obviously, as they are the roots, they are underground, they're not seeing any sunlight, they contain no chloroplast. They don't need to photosynthesize. Okay, next up, we've got the xylem cells. So the xylem is one of our transport systems in plants. And it's where we carry water and mineral ions going from the roots up to the leaves. That process is transpiration. So the xylem are very specially adapted. They are initially alive, but then they become dead cells and they make a very long hollow tube. They're covered by something called lignin, which helps provide some strength for it and means it can stand the water pressure as it's going up, and it helps support the plant stem as well, especially as plants get taller. Finally, we've got those phloem cells. These are our other transport tissues within, or transport cells that make up the tissue within plants. Now this is the one that carries all the dissolved food up and down, as opposed to xylem where it only moves water up. This is moving everything else going up and down, particularly all that lovely glucose that they've made. Now they've got special sieve tubes between each cell, which helps filter things through it. They've also got companion cells next to them, so they are mainly empty. They do contain their own nucleus, okay, but they are surrounded by cells that have lots of extra mitochondria and lots of extra ribosomes to make everything that the phloem needs. Okay, so you may have to identify specialized cells, either from a picture or from a description. You may have to describe its adaptation, which is what I've just done through all of them. Okay, you may need to describe its function as well. Okay, and you will also, especially for those of you doing the higher tier, need to explain how its adaptation enables it to perform its function. So if we're looking at the number of mitochondria is our adaptation, it enables it to perform its function. If we look at a muscle cell of providing lots of contractions, the mitochondria are able to provide lots of energy for doing those contractions. Okay, differentiation. Okay, so differentiation is where cells will change from what they are. Now in animal cells, cells differentiate very early on during embryo development. Once something has become that type of cell, that's what it is. It can't become something else again. You cannot take a nerve cell and turn it into a red blood cell. Very, very hard to do. Same way you can't take a muscle cell and put it in a lab and grow an entire human out of it as a clone. It just doesn't work. The cells can no longer differentiate past a certain point. Whereas plant cells, they can differentiate throughout their entire life. We can almost take any part of a plant, plant it in a new bit of soil, make sure it's got enough nutrients, and we will get a clone of that plant. Plant cells can differentiate, so they can become all other different types of cells, as long as it's within that plant, obviously, throughout their entire life. So, when differentiation happens, all those subcellular structures, those parts inside those organelles, will differ, and that's to enable the cell to carry out its function. So whether it's going to get more mitochondria, less chloroplast, a larger vacuole, a larger surface area, things will all change. Next bit, we're looking at microscopes. Okay, so light microscopes were first invented in the mid-17th century. Okay, so that's the 1600s by a guy called Robert Hooke. You'll come across him again in physics when we look at Hooke's law. So light microscopes are very, very good. They allow us to see live specimens, and we can magnify things up to 2,000 times, which is amazing. They are also incredibly cheap, and we've got lots of them at school as well. Now, in the 1930s, we managed to invent electron microscopes, and that goes up to 2 million times magnification. We can see very, very close to all sorts of things. We can also get 2D and 3D images, and we can see those subcellular structures in fantastic detail, which we previously hadn't been able to see. It's given biologists and chemists a great understanding about how cells can work. Only problem with electron microscopes is they cost an awful lot of money. They are very, very large and have to be kept in very, very secure places where the temperature, the pressure and everything, humidity is all controlled. But they do allow us to see things in great detail, although it does have to be dead. We cannot look at live specimens. Now, 2 words that we might need to know here. So magnification, which is how many times bigger an object appears, and resolution, which is the ability to distinguish between 2 separate points. So magnification, how many times we can zoom in and make it larger. However, that resolution is how clearly we can see the image. Can we see that 2 points are separate? The better the resolution, the better that we can see that those 2 points are separate. And you'll see there as well, we've got our calculation that we need to know for this topic, which is magnification is our image size divided by our real size. We're going to have a look at that. So, looking at our calculation, okay, we've got our calculation there. Magnification is image size divided by real size. We're going to have a go at doing a question. So we've got an image size is 1 millimeter. A real size is 25 micrometers. And we need to calculate the magnification. Now, if you want, you can have a go at doing this by yourself, and then I'll walk you through the steps of how we've done it. Okay, so first off, another thing to do is make sure all the units are the same. We cannot divide millimeters by micrometers. It doesn't work. The units go all wrong. Okay, so what we'll do first, we'll get everything into the smallest unit. So 1 millimeter is 1,000 micrometers. To go from millimeters to micrometers, I've times it by 1,000. So 1 times 1,000 gives us 1,000 micrometers. Next up, we're going to write down our equation. So we know that magnification equals image size divided by real size. Okay, and we're going to write down the parts that we know already. Now, we don't know what the magnification is. That's what we're trying to find out. We know that the image size is 1,000 micrometers, and we know that our real size is 25 micrometers. Okay, we've got most of the equation now. We're now going to put those values into the equation, putting them back into where they go. Okay, so that image size of 1,000 micrometers and that real size of 25 micrometers. So we get magnification is 1,000 micrometers divided by 25 micrometers. You put that into your calculator, or you may need to rearrange the equation, and we can calculate our answer. We get a magnification of 40 times. We're also going to do a quick common sense check and check that we're happy with that answer and that we think that's right. And we know that microscopes can be up to 2,000 times magnification. So we're looking for a number that's smaller than 2,000. Using the school microscopes, we've got magnification of up to about 100 or up to 200 times. So 40 times definitely fits in. Okay, that's our common sense check. Okay, on the right of the screen now are 6 questions which I would like you to try and have a go at. You may find it helpful to pause the video while you calculate it. Again, use those steps on the left to check you are happy with it. Okay, hopefully you've had a time to work out those answers. If you've not, please do pause the video so you can do it without seeing the answers. Okay, there are all of the answers. Okay, hopefully you have got them right. If you've had any issues with working them out, please do come and see either myself or any of the other science teachers within the department. Okay, last bit we're going to look at then is that required practical, okay, which is using a light microscope. Now, depending on feedback, I may or may not do a video demonstration of all of the practicals so you can see them being done, but for the moment we're going to concentrate on the exam questions that they may ask you. Okay, so this is one that was from your November PPEs. Okay, so the November PPEs. A microscope has a ×5 eyepiece lens. Describe how to use this microscope to observe a prepared slide of root hair cells at a magnification of ×50. Okay, this is worth 4 marks. Now, it's not a case of when I show you the mark scheme that you have to get 4 of those steps. It's that you have to have the whole thing down, and you will either get 1 to 2 or 3 to 4 marks. What you can do is spend about 5 minutes trying to write an answer of how to do it, thinking about making sure you're putting that slide on, focusing it, checking those magnifications. If you want me to have a read through of any of your answers, please do bring them in so I can read them for you and let you know where I think you've gone right or wrong. If not, I will show you the mark scheme for the answer in a minute. Okay, now you've had a chance at doing that. Here are the requirements for the mark scheme. So first you're going to place that slide on a stage. You're going to use that lowest power, that lowest magnification at first, which is our ×4 objective lens. We're going to make sure that the mirror or light is on so it passes through the slide, and we're going to move the stage as close as possible to the lens. Okay, but make sure we obviously don't touch that lens. We're going to turn the focusing knob so the slide moves away from the lens, making sure we're looking through that eyepiece, and we're going to keep turning it until the image comes into focus. And we can use our fine focus, if you've got one of those on a microscope, we don't have them here at school, to get a really, really clear image. We're then going to change our objective lens, that's that bit at the bottom that can twist round, to ×10, and make sure we can refocus it. Now, see, a ×5 eyepiece and a ×10 objective lens gives our total magnification of ×50. We do our eyepiece times our objective lens to get our total magnification, and then we can refocus using our focusing knobs, either our coarse focus or our fine focus. Okay, I hope this video has been helpful for you. If you do have any feedback, please do come and speak to me to let you know what you think could be done better or what you might prefer in the video instead. Thank you very much.